So good evening, everyone. I'm Andrew Creek. I'm a Development Officer with the New South Wales Department of Primary Industries. And I'm co-hosting this event this evening with Stephen Fellavine. You can see him there beside me on the screen. Stephen's also a Development Officer and he's based at Denton with the New South Wales Department of Primary Industries. This webinar we have this evening is an output from the Citrus Industry IPDM Extension Program that's funded by the Citrus Levy, Forward Innovation and collaborating state governments being New South Wales, QDAF and also the Western Australian Government. The Citrus IPM Extension team, we welcome you to this webinar this evening. We welcome all the people from different regions around Australia. In 2022, our extension program, we surveyed, it was quite a lot of growers, I can't remember the exact number, but anyway, red scale came up as one of the highest pests of concern for the industry. So that's why we've arrived to have a webinar with some expert presenters this evening. That being Doctor Meena Thakur, a research entomologist at Yanco, and also Rob Weppler, who I would say is a practising entomologist who monitors crops professionally here in the Riverina in the region where I am based.
Good evening, everyone. As Andrew mentioned, I am an entomologist at Department of Primary Industries based at Yanco, and today I'm going to present on California red scale life cycle and development, but I will also briefly explain what the scale insects are in general and what their feeding habits are and why it is important to monitor the populations. The scales are small sap-sucking insects in the order Hemiptera, which is also known as the order of true bugs, and there are about 50,000 to 80,000 described species within this group, and scale belongs to this group's Sternorhyncha, which includes other sucking pests, for example, jumping plant lice, whiteflies, scales, and aphids. And scales, as we can see, doesn't look like anything like any of those bugs. They are like flat, tiny bump, wart, or sac stuck to the plant surfaces, and they often blend in with the plant, making it hard to notice them. They have this characteristic protective covering that protects them from chemicals and parasites or natural enemies that we call scale, that can vary in appearance from waxy or powdery to hard and shell-like depending on the species. Except crawlers, all immature stages and adult females lack functional legs, eyes, antennae, or wings. When adults are present, because not all of the scales have adults, but when they are present, they are winged, smaller than females, and short-lived. For their feeding habits, they are sucking, piercing sucking insects, and their mouth parts is like long needle-like called stylates, which they insert into the plant tissue to feed on the sap. And the group of the insects, for example, scales, aphids, mealybug, and whiteflies, they suck the sap directly from the vascular tissue of the plants, which is full of nutrients and consists of the phloem and xylem. And there are two different types of scales that affect citrus. That is armored scale, which has hard covering that, depending on the stage of their life cycle, may be attached to or become separated from the body. And another group is soft scale, which has the protective waxy covering, but is integrated with the body throughout their life cycle. And the armored scales do not produce honeydews, but are very damaging to the plants, whereas the soft scales are not as damaging, but produces honeydew, which leads to sooty mold that causes the issues with the scales. There are 7 different types of armored scale in Australia that we found on citrus, and about 13 different types of soft scales. But among all these scales, California red scale is one of the most damaging and widespread, and we will be looking into the life cycle and monitoring of this scale today. It is widespread in all citrus growing regions and mainly attacks citrus, but can also affect about 77 plant families, which includes acacia, willows, olives, passion fruit, roses, etc. And the favorable conditions for red scale development, they prefer dry inland or coastal areas or dust covering plants and dense canopies with poor air circulation spat, because that creates the micro habitat for their growth and survival. And stressed, unmanaged plants are more prone to red scale damage than the healthier plants. All of the stages of scale insect secretes a toxic saliva during their feeding and attack all aerial parts of citrus, which include includes twigs, stems, fruits. At low level infestation, their damage can go unnoticed, but at heavy infestation, the discoloration, shoot distortion, or leaf drop is more prominent. And at severe infestations, twigs and branches may die back, occasionally leading to death of trees. And the tree damage is more apparent during the late summer or early fall season, when the scales are at high numbers and trees are under higher moisture stress. On the fruits, they cause both cosmetic and direct damage, and speckling and blemishes on the fruit is one of the main issues with their feeding. Highly infested fruit can be competed and downgraded or rendered unmarketable for fresh use, and it is a quarantine pest for the export of oranges to Korea. The life stages of red scale can be divided into two parts depending on their mobility or their maturity. So there are two immature stages and two mature stages, or two mobile stages and two types of fixed stages. So the mobile stages includes crawlers and males. Other than these two stages, there are no other stages that can move around. So once they are fixed, they remain in the same position until they become mature females. And these mature females have these reddish brown waxy covering on their top, which protects them from the chemicals or the natural enemies. And it is about 1.8 to 2 millimeters wide. The mature males has this distinctive brown line on their thorax, which is very helpful to identify them on the pheromone cards when we monitor for the adult males. Now the immobile stages are further of two different types, and it is very important to understand the difference between these stages, because that helps with the pest management decisions for the red scale. Because molting stage, which is the dormant or non-feeding stage, is very hard to penetrate and target with chemicals or parasitic wasps, whereas the instar stage, which is the feeding stage, has this separate body and the cover, and is more sensitive to the control methods. So the difference between these two stages is that when the scale undergoes molting, all the insects need to molt to grow. Body coloration changes to reddish brown or orange, and you can see there's this distinct edge where we can't separate this top cover with the body. In the instar stages, there is this separation between the cover and the body of the scale is yellow colored. And this feeding tube keeps them on the plant when we flip these covers of the instar stages over. Now coming to the life cycle, it starts with the reproducing female, which can lay up to 150 crawlers during her lifetime, and she produces 2 to 3 live crawlers per day for 6 to 8 week period. And these crawlers walk actively until they find a suitable feeding site, and sometimes they can also be dispersed by wind, and long dispersal of the crawlers is usually by wind, but there are also evidences of them riding on other insects or machinery, tools, or orchard workers. So once the crawlers establishes and start feeding, they lose their legs in a day or two, and they start to release this white waxy secretion around them. And this stage is called as white cap stage because they just look like tiny white caps. They keep growing and feeding in this stage and keep secreting this waxy structure around them. And then after the first mold, the females undergoes a different development process, whereas the males goes through a different development process. The females molt twice only, whereas the male molts 4 times to become adults. And this is it is at the third instar stage that the females mate with the males, and then they turn into gravid females, secrete another layer of wax around them, and becomes really difficult for treatments to target them. I will explain a little more in detail about the female development and show you exact picture of what they look like. So here you can see the crawlers. So once they settle, this stage here is the white cap stage. So when you flip this over, you will see the cap is separate from the scale body. So when you can separate these two, that means this is an instar stage and is more sensitive stage for any chemical or other control options. And then after first mold, they release this chitin and become hardened. And when you flip this over, you can't separate the body from the cap. And then they stay in this stage for about 4 days and secretes this waxy gray, which we call the gray skirt around them. And when you flip them over, you can separate the body from the cap. And in the second instar stage, the female body is more like a teardrop shape. So you can tell that this is the second instar. And after the second instar, undergo the second molt and produces another ring, which we call second molt ring. So this is the difference between the first instar and the second molt, or the third instar stages. And in this molt stage, again, you can't separate the top from the body of the scale. And then the second molt scale undergoes more changes and becomes third instar, where the female develop these lobes. And we can see there are two rings on the scale cover, and there's this gray skirt around them. And the females remains in this stage for as long as until the male comes and mate with them. And the width of the skirt will tell you if how long it has been for the females in this feeding stage. The longer the feeding stage, the wider the skirts. And this is at this stage, at third instar, virgin third instar stage, that the phytus species prefers to lay eggs, and this is the stage where males mate with the females. And once the mating happens, then the female secretes another waxy layer around it, becomes enclosed and really hard for the chemicals or parasites to attack. And then the life cycle goes on again. And during winters, the females overwinter in this stage and produces the crawlers in the following season. For the male development, the first instar stage is the same with the white cap stage. And after the first molt, the males become elongated with a V-shaped posterior end. And then after the second molt, they develop eyes, and then molts again and develop eyes more, and also start to produce wing pads and adigus. And then after fourth molt, they become adults. The adults lives only about 6 hours or at most a day, and they do not feed at all. And also the sole purpose of males is just mating with the females, and then they die. Because males molt 4 times, they are more sensitive to the chemicals like insect growth regulators compared to females who only molt twice, because IGRs work during the molting processes. So you need to target the instar stages compared to the molting stages when you are planning to do chemical treatments with insect growth regulators. And the minimum threshold temperature for red scale is 11.7 degrees Celsius. And the life cycle depends on the temperature and relative humidity of a particular region. But generally, there are 3 to 4 generations in southern areas and 5 to 6 in northern areas. So now, as we have understood the life cycle of red scale, it becomes very important to monitor for the most sensitive stages to plan the chemical treatments or phytus wasp releases for best management. Crawlers and whitecaps are best to target if you are going for chemical control, and especially the first post-winter or spring generations ensure better control as they reduces the population for the rest of the season. The third instar females are the most preferred stages by a phytus wasp, but they can also attack second instar, large second instar females or males under high red scale populations. Instar stages are more sensitive for IGR applications, and monitoring is also important to check the natural parasitism levels in your orchards, because if you have enough parasitism or high level of parasitism, you may not need to do any of the chemical treatments and may save your time and money. And the thing is, for scales, biological control works more effectively than the chemical control, but Rob will talk more about that later. So how to monitor red scale populations? There are different methods that we can do to monitor the scale population, inspecting leaves, fruits, or branches visually, but that can be really difficult because scales are tiny and separating individual stages in the field can be challenging even for professionals. The other method is double-sided sticky tape, but again, this can be tedious job for growers, and usually the researchers do this to accurately monitor the crawler populations. The other method is pheromone traps to monitor the adult male activity, and then we also combine the pheromone trapping data with the degree days, which I explain in a minute what that is. And there is also an online timing guide to predict the crawler emergencies or the phytus release timing that has been developed by New South Wales DPI in collaboration with Hot Innovation to help growers find the right timings for their applications. Importance of tracking male flight. So when we are planning to do any of the treatments, it is very important to track the male flights, and based on the male flights, we can identify when the virgin third instar females will be around, because that will be the ideal time for releasing the aphytus wasps. And male flight peaks can also be used to predict when the crawlers will be most abundant based on the degree days. And this can vary from region to region because of the different temperatures, but usually about 272 to 422 day gaps has been observed between peak male flights and spring crawler peaks in Sunraysia and Riverina. In southern regions, first post-winter male peak flight activity occurs between mid-September and early October, and the crawlers are most abundant in November. Now the degree days. So all the insects require certain heat units to grow from one stage to another, and degree day is a measure of those heat units. It is described as the accumulation of the average daily temperature above the lower development threshold needed for an insect development from one life stage to the next. The formula to work out this is the difference between the average daily temperature and lower development threshold, which is 11.7 degrees Celsius for red scale. And as you can see in this table here, for example, for day 1, if the temperature is 30 degrees Celsius and minimum temperature is 20 degrees Celsius, the average will be 25. And if you subtract 11.7 degrees Celsius from this average temperature, the daily degree day will be 13.3. So degree day calculation for red scale typically begins on 1st July, and spring male peaks are predicted to occur after accumulating 203 to 353 degree days from this time, and crawler peaks are predicted to emerge after 582 to 732 degree days from this time. There's this online timing guide that I was mentioning. This is called Red Scale Shiny app, and this has been developed for growers who do not use pheromone traps, and this uses degree day based on the temperature of the region and can be more accurate if male flight peak data is available. But otherwise, if you feed your region on this app, it will tell you when the crawlers will be emerging or when it will be best timing to release the aphytus based on the regional temperature data. So you can search this app by using keywords like red scale prediction tool, red scale shiny app tool, or red scale spray timing tool. I have used as an example here red scale prediction tool, and this gave me these different links, and I clicked on the first one that led me to this page, which explained a bit more about the spray timing tool. And then there's this link for the red scale shiny app. And when you click onto this will take you to this page where you can choose your region, and you can select if you have monitored male flight data or not. And it will give you prediction like this timing for the crawler emergence peaks for depending on the temperature of a particular year. I have chosen male flight data and have fed the 15th of December 2024 as the male peak flight, and it has given me more accurate data of when the crawler peaks will be during this season. So based on the average of warm or cool year, it has given me these timings for the peak crawler emergencies, and then based on this, growers can decide their treatments for red scale. So that was me. Thank you.
What determines the sex ratio of males to females when they're born? And Meena's already answered, it's about a ratio of 2.6 females to 1 male. So it's not like in humans where it's 1 to 1 is the approximate ratio. There's a few more females to males, and I gather that's because there's no need for too many males out there in the wild.
Yes, because once the females mate, they retract. See, in this slide, you can see that the virgin female has this protruding pygidium, and once they mate, they retract it back and looks like something like this here. So a male can mate multiple times, but females only mate once. So yeah, that's why I think there is no need for many males, whereas females are needed to produce more babies. And I'm not sure if there is any impact of nutrients or climate on the sex ratio, but this has been reported. This is the general standard ratio that has been studied, the 2.6 females to 1 male.
There are a number of these growth regulator chemicals, and I think you said, and please refresh us, that you said it affects it works better on the males and the females, or Could you please discuss that again whilst the slide's up there? And what type of chemicals are they? Can you name them?
So insect growth regulators, for example, buprofezin based one, or pyriproxyfen is one. Yes, pyriproxyfen is one, buprofezin is one. So those are the ones that are usually used. And what they do, they stop the molting process of the insect. So they are more effective on these instar stages, the stages between the molts. So when you can separate the scale body from the cap, that means they are in that instar stage, and this will be the most sensitive stage for those chemicals to work, because once they are in the molting stage, for example, here in the females, it's more visible here. This one is the molting stage where the female body is attached to the cap. It is very hard for the chemicals to penetrate and affect the females at this stage, or the males at this stage. So the instar stages are the more sensitive stages, or the crawler stages, and the white cap.
So when they're changing from one instar to another, that's when the chemical does its work. And I gather now, understanding a practical application, if you spray one of these chemicals, will it hang around long enough in the tree where, let's just say, it could be, I don't know, a week between one instar and the other, or whatever it is? Will it hang around long enough to catch those different wait around until that stage comes out? Or will the chemical just be there for a couple of days and it'll only catch those scale that are changing at that time and miss all the others completely?
And it will vary from chemical to chemical, but generally, because as I said, when you spray, for example, if you are spraying the white cap stage, and where the instar, the body is separate, the chemicals can penetrate and reach the scale body. Whereas if you spray it on this stage, which is molting stage, that there will be a hard chitin layer around the female, all around, not just on top, but also on the underside of the body. And then the chemicals can't just penetrate. It is more like that chemical can stay there for a while, because most of the chemicals has longer residual periods. But then, because this body will be then separated from the female, and then she will then release another layer, so she will be pushing another layer onto the top layer. So I do not think that will work. The first application would work. You will need to do another application to reach to the female body. Whereas if you are talking about crawler stage, the crawlers will be crawling around and they can pick those chemicals, so they can get impacted. But once they are in these molting stages, it's really hard to reach the bodies underneath.
So look, I invite Rob Weppler, an entomologist in the Riverina. Rob is also a member of the Citrus IPDM extension program team. Rob is monitoring a lot of demonstration sites for me locally here in the Riverina, and he's also reviewing some of our many publications. I think we have around 30 fact sheets, we've got posters, and we're working on a field guide as well. Thank you.
A little bit about my background. I'm originally from California, where I studied under Joe Morris and Bob Duck at the University of California, Riverside. Since 2006, I've been working as a citrus entomologist in the Riverina of New South Wales. I want to acknowledge all the people on this screen, as they have greatly influenced my understanding of red scale, its biology, biological control, and management. I feel a bit funny talking as an expert with a lot of these people here in Australia, because they really know their stuff. Anyways, I also want to acknowledge these publications at the bottom. Some of the photos I'm going to show are from these publications, but they're a great source of information. So Meena's already talked about damage. Actually mentioned, red scale can cause twig dieback and even kill a tree. This tree kind of shows that it's got a bit of twig dieback, and it's a little bit hard to see, but that trunk is completely covered by red scale. However, this is very unlikely to occur unless the natural enemies have been eliminated by the use of pesticides. The main damage that you would all be familiar with is infestation on fruit, leading to a downgrading of fruit quality. This fruit is obviously heavily infested, but any fruit with more than 10 red scale present is likely to be downgraded. Meena went through the monitoring quite well, so I won't spend too much time here, but you do have this pheromone sticky traps where you can monitor male flights. We haven't found that to be particularly useful, but it is useful from the standpoint of if you determine when the peak in male flight is, then you can use those degree days to predict when certain scale stages will occur. So it is useful for that. We find visual inspection of fruit to be the best method. And the difficulty with it is that it's time consuming, but it gives a very good indication of scale activity. You need to be able to recognize live versus dead and parasitized scale, which can be very difficult to assess in the field. And again, it's because, as Meena showed, the body is underneath that waxy cover, so you can't see that directly unless you pull that cover off. Bin checks is another way. And I think probably all growers do this, whether formally or informally, but it's just have a look at your fruit before you send it in. If you're seeing a lot of red scale, it's too late for that season to do anything about it. But that's kind of an indication that, okay, we need to rethink our strategy for the following season. And also parasitism assessment. This is best done by collecting infested fruit and checking with a microscope back in the laboratory. This is more important where there are higher levels of red scale and growers are relying on biological control. I guess an important part of that monitoring is recognizing it and differentiating it from other things that might look similar. On the left is the male scale, like Meena showed, but on the sticky traps, you'll also get a Phytus wasp, which are similar in size and similar in color. But if you look with a hand lens or a microscope, they look quite different. And as she pointed out, the scale have this black or brownish line across their back. And that's a giveaway that it's a scale and not an Phytus wasp. But you don't want to be counting a Phytus wasp when you're trying to count scale on this card. And then on the right is just a range of scale stages. And we often see multiple stages of scale at the same time in the field. And I've just put an insert here where I've pulled one of the covers off to show the scale body underneath it. And that's important if you're monitoring, but again, difficult in the field because you need to be able to look under and go, yes, that scale's alive or dead or parasitized. Okay, so there's a long history of trying to eradicate red scale with insecticides. Nearly all of the insecticides on this page worked well initially, but due to resistance developing and the very toxic nature of these chemicals to natural enemies, they eventually failed. Most of these insecticides are no longer available for use in citrus, and those that are not very effective against red scale, but still very toxic. And just interesting, on the back of this photograph was written, raising a tent to cover a citrus tree in order to fumigate with cyanide gas. Sometimes men as well as bugs were killed. So it just kind of shows that for a long period, people have been going to great lengths to try to get rid of red scale. And I think someone asked about the names of the chemicals, so that's there. But we now have a number of newer pesticides available to use against red scale. These products tend to be softer on natural enemies, but are still toxic to some. They also have a narrower range of activity, meaning that they control some pests, but not others. Some are very effective, but all of them have their drawbacks. And red scale is best managed rather than controlled. And if you think that you're going to spray something and completely get rid of it from your orchard, it's just not going to happen. It's unrealistic. So let's look at management options. I'll talk a little bit more about aphitis and maiden disruption later, but the big difference I wanted to highlight here is that between these options and the chemical options is that they only affect red scale and they're not toxic to any other species. And there's also very, very little chance of resistance developing against them. The oil sprays can be very effective if applied to young scale. I think someone was asking about oil sprays. Yeah, they can be effective, but you need to be really careful about phytotoxicity. And because oils work by suffocation, they'll kill most insects that they get adequate coverage on, particularly the soft-bodied insects. But because there's no residual control, the timing's really important. And that's a good thing in terms of their effect on natural enemies, because even though they kill most natural enemies that they contact immediately, there's no residual control after the fact. So they're not that hard on the natural enemies. Pyrotetramat or Moventa, we see good control of most red scale stages with this chemical, but I've heard that it is not as effective against scales on the stems and woody parts of the tree. And that kind of plays out with what we see from one season to the next with red scale. Movento is soft on parasitoids and predatory beetles, but it's toxic to predatory mites. The insect growth regulators, I don't have much experience with Applaud against red scale, but Admiral is highly effective, particularly if it is applied to the first instar scale. And again, like Meena was explaining, you want to get it just prior to it molting to that second stage. That's the ideal time. And it's fair, both the insect growth regulators are fairly soft on most natural enemies, but they're very toxic to predatory beetles. And this can cause problems with things like cottony cushion scale that rely on in Cottony Cushionscale's case, the Vidalia beetle for its control. There's also been resistance developed in red scale to admiral, and that's developed in some parts of the world. We haven't seen that here in Australia, but we have seen resistance in whiteflies to admiral in Australia. A couple other options, the neonicotinoids, I've lumped them here for their general traits, but in terms of efficacy, they vary from product to product. We have not seen them to be great as a group on red scale. Confidor is probably about the best, but even with Confidor, it's not thought to give good control of scale in on the woody interior portions of the plant. And we often see good control for a good portion of the season with Confidor, but then late in the season, all of a sudden we started getting a lot of scale moving back to the fruit. The neonicotinoids are toxic to most natural enemies, particularly if applied foliarly. There's less impact if they're applied through the irrigation, but we still see some outbreaks of two-spotted mites and lepidopteran pests like light brown apple moth and loopers following their use. So even though they're not directly killing the things when you put them to the water, they are affecting the natural enemies and causing some other problems. Okay, the organophosphates and carbamates, I've also grouped them together for their similarities, but again, each individual product varies quite a bit in how effective it is against red scale. What tends to happen with them is they kill some scales, but we tend to see a resurgence of both red scale and other pests after their use. Particularly mealybugs is one that we often see sort of flare up after we've used those sort of products. And they're very toxic to nearly all natural enemies. Okay, so chemical control of red scale is challenging. And Meena mentioned this in her talk as well, but they're sedentary and they're covered by wax, making it difficult to get good coverage of the insecticide. They're also quick to develop resistance to insecticides. All of the foliar treatments that we've mentioned are more effective when targeting young scale stages rather than adults. And I guess probably if you take one thing away from my portion of this talk is that if you're going to spray red scale, you really need to spray well. And that means high volumes and traveling at slow speeds. You need excellent coverage of the interior parts of the tree and at the top part of the tree. And you also need to choose the right product for the situation. Some of the products will only affect certain stages. Some of them will cover a slightly bigger range. Good application lowers the potential for resistance to develop, and poor control can be worse than doing nothing at all. And that's partly because if you don't get good control, you've still got the scale, but you've knocked out some of your natural enemies. I'm not going to go through the life scale cycle of red scale again, but I just wanted to put it in the context of our control options. So with oil, you really need to target these very young stages, the crawlers and the whitecaps. You can get a little bit of control of older stages with good coverage, but really it's these younger stages that the oil applications are good against. And the same is true for the broad spectrum insecticides, the organophosphates and the carbamates. But again, with really good coverage, you will see some older scale also dying. For best results with the insect growth regulators, the Admiral and the Plod, you want to target the first instars. So you want to get them before they molt to that second instar, and Meena explained why that is, so we don't need to go back through that. Movento we find to be pretty good on nearly all the growth stages, but we often see mature females that are already producing crawlers. Those crawlers will still emerge, come out onto the fruit and settle, and for a period of time they still look to be alive, but usually they don't survive. Aphytis prefers to target the unmated third instar female scale, so this stage here, and Campariella, another important parasitoid of red scale, also prefers the third instar scale, but will also attack mature female scale. Meena talked about degree days a bit. This is just the data that I've recorded from here in Griffith since I've been here, and we find this a little bit useful early in the season, but not so much later, and I'll explain why in a minute. So this chart shows the accumulation of degree days. The thicker black line in the middle is the average from all these years. And then each other line going up is just a different year. The bright green line shown here is this current season. And these blue lines that are crossing are when I would predict the peak in crawler emergence to occur. Like I said, this works fairly well for the first generation, but because red scale produce 100 to 150 crawlers, with only a few coming out each day, and red scale can develop at different speeds based on the variety and where the individual scale has settled, whether on leaf, fruit, or stem, subsequent generations increasingly overlap. So often early on, we'll see discrete generations and we can say, okay, here's the peak in crawler production, and here's the stage we want to target, and we can apply something at that stage. When we get to these later times, we see all sorts of growth stages. The other thing that's been interesting in recent years is that red scale often gets sort of synchronized over winter because low temperatures affect the molting stages and the younger stages more than they do the older stages. So we often come out of winter with mostly older stages of scale. When we've had these mild winters like we've had for the last few years, not so much this year, but the previous years, we see all ages of scale right from the beginning of the year. So I think the degree days can be useful, but it's probably better to also be looking at fruit to really time your scale spreads. All right, I want to talk just a little bit about mating disruption because it's a fairly new option in Australia. Male red scale are attracted to females by a chemical signal called a pheromone. Mating disruption floods the area with a synthetic version of this pheromone, which reduces and delays males from mating with the females. And this has two benefits. First of all, the unmated female scales do not produce crawlers. But also, by delaying that mating, that is the most susceptible stage to parasitism. So it allows the Aphytis and Campariella to do more work and get higher levels of parasitism. And in research trials in Spain and California, they've shown mating disruption to be very effective at reducing red scale infestation. So Checkmate CRS was registered for use in Australian citrus in 2022, and it comes in terms of these dispensers. So you get a pack of these dispensers and you hang these in the tree at a rate of 450 per hectare. These dispensers then emit sufficient pheromone for about 154 days to interfere with the male's ability to locate the female scales. In high red scale populations, you need to apply an insecticide prior to using mating disruption. And this is because once a male is in the vicinity of a female, they use other cues to locate and mate with the females. And at very high red scale densities, they just bump into each other, and finding the female is less of a problem. In California, best control was achieved when placed into the orchard prior to the first flight. So there's another good use of those degree days. You can say, okay, here's where we expect the first flight to be. Let's get the mating disruption in before that. In all their trials, they got around 75% reduction in severe infestation. But when they did a Moventa early to lower the population and then put out the pheromone dispensers before the first flight, they got up to 95% reduction. So quite effective. And I think this has a real place with us in managing red scale because it's compatible with both biological control and with chemical application. So we'll talk a little bit about the natural enemies. The most important natural enemy at controlling red scale population is the Aphytis wasp. Two species predominate, Aphytis malinus in the southern areas and Aphytis lignanensis in the northern and coastal areas. Both species are available for augmentative release to boost the natural population. They come in cups of about 10,000 wasps and should be released at least 2 or 3 times at a rate of 25,000 per hectare. More releases of lower rates can also be effective. I also just want to draw your attention. So that's the adult wasp here, and this is the pupae of Aphytis. So if you're monitoring red scale and you pull the cover off, like has been done in this photograph here, and you see this sort of pupae, and sort of the giveaway with these fecal pellets, if you see that, that's Aphytis and not red scale. And so that's a good thing. We also see parasitism by Campariella and bifasciata and Incarcia citrina. Campariella is particularly important at controlling yellow scale, which most people haven't heard of, but also occurs in our orchards. But because it's indistinguishable from red scale in the field, it just gets lumped in with red scale. Lacewings do feed on red scale. How important they are at controlling the population, we're not sure. But they're very, very prevalent in our orchards, particularly in spring. And just in the bottom here is a picture of an adult green lacewing. They don't actually feed on the scale. In fact, they don't feed on other insects at all. But here are the eggs, and it's not a great picture, but inside here, that's the larva, and that's the stage of the lacewing that feeds on red scale. And it's a little eating machine. It wanders around, finds any soft-bodied insect. In the case of scale, it goes up to the scale, and we've actually seen them doing this. They push their mouth parts up, sort of lift that scale cover up a little bit, put their mouth parts into the body, and then just suck the fluids out of it. And then we'll put the cover on their back to disguise themselves. They're a very good predator, but I'm not really sure how they would help with red scale control. You probably wouldn't rely on them entirely to control red scale. There are also a number of other predators. So we'll just keep on the bottom for now with the predatory mites. We see a number of predatory mites in the orchard. The members of the family Phytosaiidae are the most common, and there's a number of different species of them in our orchards, but we have also seen anistids and bedelids feeding on red scale. And then in the top row are some ladybird beetles that feed on red scale. So we have Rhizobus lopanthe, or it's commonly called Lindorus. That's common in our area. I'm not sure how common it is in the northern areas, but the other two, Halmus and Chalicoris, are more common in the northern areas of Australia. We have, on one occasion, I've seen Chalicoris in the Riverina. So predators help with red scale control, but probably are not as important as the parasitoids in regulating the population, at least in the drier inland areas. Andrew Beattie did some really good work on red scale on the coast and found that those ladybird beetles were extremely important in regulating the population there. I've added this slide to make the point that you should not manage red scale or any other pest in isolation. You need to think about the entire pest complex and the natural enemies that help to regulate them. Here I've listed a number of pests that we regularly see in the Riverina. Most of these are not economically important, but could be if the things that control them are disrupted. DPI has recently put out a great resource that lists the pests and their scientific names of their natural enemies. You can use this to search the internet for more information about a particular predator or parasitoid. It also has a table showing the toxicity of pesticides to natural enemies. I think this can be a great tool, to help people, one, work out which pesticides to use, but also to learn a bit more about these natural enemies. In terms of integrated pest management, red scale is difficult to control chemically. Without the assistance from biological control agents, red scale quickly becomes unmanageable. Right now, we are in a period where we have some insecticides that are working very well and keeping fruit mostly clean. But if we learn from history, it is likely that this situation will not persist indefinitely. To prolong the effectiveness of the current chemistry, it is important to integrate other management options, such as augmentative releases of Aphytis wasps, and to employ mating disruption. It is also important to preserve the predators and parasitoids that naturally occur in your orchards. They provide a free service that goes unnoticed until they're gone. So that's all I have. Thank you for your time, and I hope I've given you some useful information.
It was about ants. Does ant control, if you control your ants in the orchard, will that help stop red scale problems?
Yeah, definitely. Ants are probably more important for some of the other homopteran insects because they produce honeydew, but ants tend to protect these insects from their natural enemies. And we've definitely seen more red scale where there's problems with ants, particularly the Argentine ants. So yeah, but not only with red scale, but a lot of your pest ant control will help greatly. Yep. There was a question about how many growers buy Aphytis to release. I don't know about growers in other areas. In my area, there's not a lot of growers that are purchasing Aphytis for release. I think that's probably a bad thing. And I think part of that is because we now have some chemicals that are working very well, people don't want to spend extra money also on releasing Aphytis. But, at least in the Riverina, we have a lot of juice fruit, and you can put up with a bit of scale in there. And I think it's really a good idea for growers, not just for a red scale and a Aphytis, but to try to maintain their natural enemies in those juice varieties so that if they are spraying in their export varieties and their fresh fruit varieties, they still have a population of the beneficial insects that can move back over later on. There's two insectaries that produce Aphytis, and because fewer people are purchasing them, it's getting questionable whether they're going to continue to do that. And I hope they do, because it provides a real service to our industry. But they're obviously in business, and if they're not going to make money from it, then they're going to stop doing it.
So the question, Rob, in the chat was, what is the threshold for juice for red scale? You mentioned that I don't really like thresholds because it depends on the season. It depends on how much natural enemies you have there. So there's a lot of things. So I don't really like hard, fast thresholds. But one, you can tolerate a fair amount of scale in juicing varieties because it's mainly a cosmetic pest. If it gets really, really bad, it can reduce the yield and cause twig dieback and that sort of thing. But that's pretty rare. If you still want to keep it down, I guess we kind of use around 20% is that we might start thinking about doing something. But in some of the orchards that we look at that are juicing varieties, we let it get higher than that and we don't do anything. We sometimes use some oil just to knock it back a little bit. But if we know we've got good parasitism and things haven't been disrupted by pesticides, the parasites and predators will often bring it under control. And
It's a bit scary because with us out there checking, we still see a lot of scale on fruit and the numbers can be pretty high. But come harvest, the fruit looks pretty good. It doesn't mean they're perfectly clean. Like for your fresh fruit varieties, you know, the tolerance now is so low that we really are trying to get almost to no scale on the fruit. But with juicing varieties, we can get, they end up coming pretty clean by the end of the season if you have good natural enemy activity.
I'd like to acknowledge that this event today has been by your Citrus Levy, Horde Innovation Australia, and also the collaborative governments who have contributed to this extension program. I'd also acknowledge Biological Services and River IPM for their contribution to that extension program as well. You can see there a QR code. You can scan that later on when we publish this edited presentation. It'll take you to the IPDM extension program section of the DPI website, where we now have 8 videos published. There's almost 30 fact sheets and lots of other information about pest and disease management.